Sea urchin is made up of 7 main parts. It has a water vascular system, a mouth, anus, and digestive tract, a nerve ring, gonads, spines, and tube feet. The water vascular system helps circulation in sea urchins and is involved in the movement of sea urchins by pressing the tube feet. The mouth is made up of 5 bone-like pieces that help scrape at kelp and other food sources. The digestive tract takes up the majority of the sea urchin's insides. After being digested, the waste is released from the opening on the top of the urchin. The nerve ring located above the mouth is used in place for the brain due to the sea urchin's lack of brain. The reproductive organ of the sea urchin, near the top of the opening where the waste comes out, releases the sea urchin's sperm or eggs depending upon whether the urchin is male or female. The spines of the sea urchin is used for protection against predators and can also be used to burrow into rock for added protection. The tube feet of the sea urchins are small and weak. They are powered by muscles in the feet. Although they are weak, their sheer numbers make them effective. Sea urchins are peaceful nocturnal creatures, as long as they are not bothered by predators and as long as they are not kelp. Sea urchins are found in salt water. They prefer shallow depths and tend to stay near rock pools and coral reefs. The sea urchin's main predator is a seabird. It will catch the urchin and drop from a high height and drop into a hard surface. This breaks the urchin's outer shell. Other predators of the sea urchin include fish, like the parrotfish, who are good at chewing hard objects. One of the main predators of sea urchin is the sea otter. When they catch a sea urchin, they lie in their box and smash the urchin against the rock.
Sea urchins have 5 gonads. These are where the sperm and eggs are made depending on the sex of the sea urchin. When the sperm or eggs are ready to be released, muscles help push them out.
Sea urchins are members of the phylum Echinodermata, which also includes sea stars, sea cucumbers, brittle stars, and crinoids. Like other echinoderms, they have 5-fold symmetry, called pentamerism, and move by means of hundreds of tiny, transparent, adhesive tube feet.
The cell cycle is a series of events taking place inside a cell to prepare for DNA replication as well as cell division. There are 2 main phases of this cycle, interphase and the mitotic phase. Both of these are divided into smaller sections. Interphase is composed of 3 subphases called growth 1, synthesis, and growth 2. Cells in growth 1 phase prepare for the next step, synthesis. In G1, cells create macromolecules such as proteins, and then later on they create enzymes needed in synthesis or S phase. To better understand DNA duplication, here is some information on its structure. DNA molecules are shaped like double helixes because they are anti-parallel. Each strand is made up by a series of nucleotides. Each nucleotide has 1 pentose sugar, a phosphate group, and a nitrogenous base. The 4 bases used in DNA are adenine, thymine, cytosine, and guanine. Adenine can only bind with thymine, and cytosine can only bind with guanine. The bases are separated into 2 groups, purines and pyrimidines. Adenine and guanine are purines, and thymine and cytosine are pyrimidines. In DNA replication, several enzymes are used, the first enzyme being DNA helicase. Helicase's job is to unwind the double helix and break apart hydrogen bonds between the nucleotide bases. Single-strand binding proteins support the open helix so it will not close. Next, topoisomerase will act as the backbone of the molecule. Of the 2 DNA strands, there is a leading and lagging strand. The leading strand is the one that goes to 3 to 5 prime. Because DNA polymerase III reads in that direction, the nucleotides are laid down continuously. On the lagging strand, polymerase III has to periodically lay down fragments of nucleotides called Okazaki fragments. RNA primase lays down RNA nucleotides to make polymerase III's job easier. DNA polymerase I takes away any RNA nucleotides and their places are filled by DNA nucleotides when polymerase III comes back. The last enzyme to be used is DNA ligase. This enzyme binds the Okazaki fragments together using phosphodiester bonds. If all goes well, there will be 2 separate and identical molecules of DNA. However, there are malfunctions in DNA replication. 1 in 1 billion nucleotides is mismatched or mutated. To repair these mutations, a thymine dimer is used to displace the mismatched nucleotide. An enzyme called nuclease cuts out the portion of damaged strand, and the nucleotides are replaced by polymerase III. Ligase then seals the free end of the new DNA to the old DNA. When the DNA is finally copied and ready, cyclins are made. This is a protein that, if combined with cyclin-dependent kinase, will create mitose promoting factor, or MPF. Without MPF, cells would not be let into the G2 phase. MPF helps later on in mitosis by phosphorylizing other needed enzymes. The G2 phase is a period of rapid growth and protein synthesis. After G2, the cell begins mitosis, the shortest phase of the cell cycle. Like interphase, mitosis is divided into subphases: prophase, metaphase, anaphase, and telophase. Prophase begins with the chromatin condensing into chromosomes. Centrioles, organelles that aid in cell division, move to opposite poles of the cell. Spindle fibers shoot out of the centrioles, and the nuclear envelope breaks down. The next subphase, metaphase, is when the spindle fibers help coordinate the chromosomes in the middle of the cell. In anaphase, the duplicated chromosomes called sister chromatids separated the kinetochore. The kinetochore is where the centromere of the sister chromatids attaches to the spindle fibers. The separated sister chromatids are pulled to each end of their centromere by motor proteins. During telophase, the last subphase of mitosis, the chromosomes arrive at different poles. Each daughter nuclei are formed around the chromosomes, and the chromosomes are no longer visible under a microscope. In animal cells, one would be able to see the constriction of microfilaments around the cell called the cleavage furrow. The final part of the cell division and cell cycle is cytokinesis. Cytokinesis is the splitting of the cytoplasm. After this, there are two daughter cells. Throughout the whole cell cycle, there are checkpoints. The three major checkpoints are after G1 phase, at the end of G2 phase, and during metaphase. The cells that do not pass the G1 checkpoint are sent to a different phase outside of the cell cycle called G0. Cells go through normal metabolic processes in G0. Many cells spend the majority of their lives in this phase. Cells that replicate infrequently, like liver cells, stay in G0 until replication is needed. At the G2 checkpoint, the ticket to pass is the right amount of MPF. At the metaphase checkpoint, all of the chromosomes must be aligned in the center of the cell for the cell to pass.
You flip the syringe to make sure that there's no air bubbles. You push it up until the solution's coming out the top. Okay, so here's the sea urchin. Okay, here's its mouth in the middle, and its other opening is on the other side. So all the gametes, the eggs and the sperm, come out the bottom side, and we inject into the top. So you put the syringe in the soft tissue. I'm sure it does. And then you slowly inject it.
Is it moving?
It moves really slowly. I can feel these spiny things moving a little bit.
Yeah.
And then, once it's injected, you
That's a lot.
be very careful that you do not cut yourself with this. And then what you do is you rock the sea urchin a little bit to move all those chemicals around. And you put it on a cup and you wait a few minutes and they will drop the either sperm or egg into the cup. It's too big for you.
Oh no.
And then they drop them in there, and so eventually you end up with a full cup of either eggs or sperm.
Drop a full cup?
And how you know that the eggs are actually orange, and the sperm are kind of like cloudy white like that. Okay, does anybody else want to come get a picture of the injection just up close? I can inject them again if you guys want to get another. Thank you. He will reenact it for you.
Procedure. Pick up the sea urchin while wearing gloves. Handle them carefully. Hold your urchin while injecting 1/10 to 2/10 milliliter per inch, a total of 2 injections. Gently rock or shake the urchin for a few seconds to mix the KCl solution inside the urchin. Do not shake them too hard or you will kill the urchin by ripping apart the delicate internal membrane. Place the male urchin mouth side down on a sterile dish. This dish should not have water on it. In a minute or two, you will see the white sperm appear on the surface of the urchin. Collect the sperm in a pipette and place it in a cuvette. Keep concentrated just until you need it. You can place the tube of sperm in the fridge and it will keep up to 5 days. Any eggs that you do not immediately use can be left in a beaker with salt water. Materials: 0.5 mole potassium chloride solution, seawater, fertile urchins, 5 cubic centimeter syringe with 25G needle, plastic pipettes, microcentrifuge tubes to store sperm, sterile beaker, microscopes, microscope camera.
To extract the right amount of sperm and egg from the sea urchin, inject both the male and female sea urchins with 1 to 2/10 of a milliliter per inch of potassium chloride. The sperm or eggs, depending on the gender, will be released out of the top. Collect the sperm and eggs in separate cups. Mix the sperm and egg in a petri dish for fertilization to start.
Radioholoblastic cleavage. Sea urchins exhibit radioholoblastic cleavage. The first and second cleavages are both meridional and are perpendicular to each other. That is to say, the cleavage furrow passed through the animals and vegetal poles. The third cleavage is equatorial, perpendicular to the first two cleavage planes, and separates the animal and vegetal hemispheres from each other. The fourth cleavage, however, is very different from the first three. The fourth cells of the animal tail divide meridionally into 8 blastomeres, each with the same volume. These cells are called mesomeres. The vegetal tail, however, undergoes an unequal equilateral cleavage to produce 4 large cells, the macromeres, and 4 smaller micromeres in a vegetal pole. As the 16-cell embryo cleaves, the 8 mesomeres divide to produce 2 animal tails, AN1 and AN2, one staggered above the other. The macromeres divide meridionally, forming a tier of 8 cells below AN2. The micromeres also divide, albeit somewhat later, producing a small cluster beneath the large tier. All the cleavage furrows of the 6th division are equatorial, and the 7th division is meridional, producing a 128-cell blastula. Blastula and blastocoel. The blastula stage of the sea urchin development begins at the 128-cell stage. Here, the cell forms a hollow sphere surrounded by a central cavity, or a blastocoel. By this time, all the cells are the same size. The micromeres have slowed down their cell division. Every cell is in contact with the proteinaceous fluid of the blastocell on the inside with the hyaline layer on the outside. At this time, tight junctions unite the once loosely connected blastomeres into a seamless epithelial sheet that completely encircles the blastocell. As the cells continue to divide, the blastula remains 1 cell layer thick, thinning out as it expands. This is accomplished by the adhesion of the blastomeres to the hyaline layer and by an influx of water that expands the blastocell. The fate map of the sea urchin's embryo was originally created by observing each of the cell layers and what its descendants became. More recent investigations have refined these maps by following the fate of individual cells injected with fluorescent dyes. These studies have shown that by the 60-cell stage, most of the embryonic cell fates are specified. These studies have shown that by the 60-cell stage, most of the embryonic cell fates are specified, but that the cells are not irreversibly committed. In other words, particular blastomeres consistently produce the same cell types in each embryo, but these cells remain pluripotent and can give rise to other cell types if experimentally placed in a different part of the embryo. The animal half of the embryo consistently gives rise to the ectoderm, the larval skin and its neurons. The veg1 layer produces cells that can enter into either the ectodermal or endodermal organs. The veg2 layer gives rise to cells that can populate 3 different structures: the endoderm, the coelom, which is the body wall, and the secondary mesenchyme, which is pigment cells immunocytons, and muscle cells. The first tier of the micromeres produces the primary mesenchyme cells that form the larval skeleton, while the second tier of micromeres contribute cells to the colon. During the 2-cell, 4-cell, or 8-cell stage, what would happen if the cells removed or damaged? Well, what would happen, depending on what the cell is destined to become, as we just discussed in the fate map, it would have some various effects on the sea urchin later in life. If you have a cell that's destined to become a mouth, the mouth of the sea urchin, okay, and that cell is damaged, later on in the development of the sea urchin, and eventually when the sea urchin is an adult, if it lives to be an adult, the sea urchin is going to have a mouth that does not work as well because it is damaged, because from birth it was damaged. Alright, Cray, I'm done!
What you will see next is early urchin cell development.
Gastrulation is a phase early in the embryonic development of most animals. During this stage, the single-layered blastula changes into a 3-layered gastrula. It looks like this. Right here you see the blastula, and here you see after gastrulation the gastrula has formed. In the blue you see the outside ectoderm, the yellow in the middle is the mesoderm, and the green near the inside is the endoderm. Gastrulation takes place after the cleavage and the formation of the blastula. The ectoderm ends up changing into the epidermis and the nervous system. Also, the mesoderm, which is in the middle, changes into muscle and cartilage and things like bone and blood. Finally, the endoderm changes into the digestive system and the respiratory system, and organs in the digestive system like the liver and the pancreas. Organogenesis is the process where the ectoderm, endoderm, and the mesoderm start to develop the internal organs of the organism. Usually this starts in humans between the 3rd and 8th week in utero. Deuterostomia is one of the major groups in the animal kingdom, and it includes the echinoderms like starfish and sea urchins, but we are also deuterostomes. And what deuterostome means is just basically mouth second. So what happens is the anus develops first from the blastopore, and later after it develops, the mouth develops from an opening in the embryonic gut. Once sperm penetrate the membrane of the egg and the flagellum is detached, powerful enzymes eat away at the egg so the sperm can fully enter. This is called the acrosome reaction. The sperm may trigger egg activation via the interaction between a sperm protein and an egg surface receptor. It is possible that a receptor is activated by the sperm binding, which activates a tyrosine kinase, which then activates phospholipase C. The fast block to polyspermy, which is when multiple sperm fuse with one egg, is an electrical defense mechanism. The resulting potential of an egg is negative 70 megavolts. After contact with sperm, an influx of sodium ions increases the potential up to positive 20 megavolts. The slow block to polyspermy is a mechanism triggered by a calcium increase. The reaction releases enzymes to release the sperm from the egg. The fertilization envelope is formed after the sperm unites with the egg. It creates a glycoprotein membrane called zona pellucida that blocks polyspermy. Now here are the bloopers.
The sea urchin's main...
What?
Jacob!
Get out of the picture! You're just like, look at this! The sea urchin's main predator is the seabird. It will catch the urchin and drop it from a high height onto a hard surface. This breaks the sea urchin's outer shell. Jacob?
I can't walk. Catch the urchin and drop from a high height and jump into a high surface. I got that wrong. It's hard. That wasn't my fault. Hey, look.
It was your fault.
The sea urchin's main predator is the seabird. It will catch the urchin. God, I said urchin. I'm getting a lot done today.
God dang it, Jacob.
Started in the water, now I feast. No matter where you look, you see me. Started in the water, now I feast. No matter where you look, you see me. I'm a Euglena, yes, a Euglena. I'm a Euglena, yes, a Euglena. Started in the water, now I eat. No matter where you look, you see me. Swimming in the water causing problems. Got chloroplast, but I'm not a plant. Zipping through the water with my flagellum. Making urchin pop, dip like a pendulum. Autotrophic and trop got the best of both worlds. I'm sitting here in my petri dish. Survival of the urchin is just a wish. All the urchin I see. Survival of the fittest is all about me. All these parts I'm known as even. Showing all the world of the urchin Steven. Hashtag my booty.
True. Based on a true story. If the cell is destined to become the mouth of the sea urchin, if a cell is missing in the very beginning of its life, it'll have a gimpy mouth.
What?
During the 2-cell, 4-cell, or 8-cell state. Alright. Gray, what do you think would happen? You don't know? Alright, let's teach these fine, Come on! Do you know? No? Neither. Actually, I do know. God